So our first presenter today is Jake Faris, and I remember Jake 2 years ago coming up from his breakout group report and suggesting we need something like a data-enabled life sciences alliance, and here we are 2 years later.
Thank you. Okay, a lot of folks part of that genesis, and as Jean noted earlier, all of that was that circumstance was made possible by the support of the National Science Foundation, which is appropriate to note because I'm going to talk about our proposal to the National Science Foundation. But first, I just have to say, following Larry Smarr, that's a nice spot to be in. But I did write down something that we at DELSA should feel very good about. He said, "My great hope lies with you." And that certainly warms my heart and whets my appetite for the future. Our future, though, is challenging. And this image here is one of a cajillion images we could throw up here to evoke the challenges of sustainability. And that's what this proposal is addressing. We, the all-inclusive global we, in fact, of course, created this challenge of sustainability. And it's a corollary, a little less obvious, but I think certainly true, that we, collectively, can successfully respond to this challenge. But in our view, will take outstanding science and significant actions and investments based on solid scientific findings. So we will need to build new scientific projects using massive complex data and interdisciplinary international teams. We will need a new generation of scientists whose training prepares them for this new kind of work. And we'll need policymakers and a general public who are informed and supportive of sustainability science and who will support policies and investments based on research findings. I would submit we have a lot of work to do. Our National Science Foundation Research Coordination Network proposal builds on the existing platform of DELSA. I think DELSA was almost unconsciously designed to make possible things of this nature. It is, of course, an international research network, and we want to build on what DELSA has in the following way. We will support and strengthen sustainability science efforts already underway both within DELSA and external to DELSA. We will help connect diverse scientists and scientific teams so that they can explore new approaches to sustainability science and design novel research projects for high impact. And we will develop communication tools, briefings for policymakers, TED Talk-like videos, mini online courses, broad-based citizen scientist projects, and others to help build that base of understanding and support that we're going to need both at the policy and the general public level. So the basic structure of this is a network with some key nodes. You have two categories of nodes. There are research nodes and support nodes. I'm just going to race through the list of prospective nodes. This has not yet been fully assembled, of course, but we have in mind the following to be included: the National Ecological Observatory Network, the National Evolutionary Synthesis Center, iPlant Collaborative, North Temperate Lakes Long-Term Ecological Research, the DELSA project, synergies between environmental, ecological, and biomedical sciences, Earth Microbiome Project, and others. Those are prospective research networks. The prospective support nodes include the international outreach DELSA activity already underway, the information resources support element, and the training and education support element, all parts of all initiatives within DELSA. In addition to that, the communication avenue support element and partner organizations support element. I know those are just titles and categories, but there's a lot of work either underway or anticipated underneath each of those. We also have an excellent international steering committee composed of Samir Faraj from McGill, Karina Gries, University of Wisconsin-Madison, our good friend Jean Kolker at Seattle Children's Research Institute, Rob Knight at the University of Colorado Boulder, whose work is cited in, by the way, in the Michael Pollan article, which I hope you've already logged into your laptop, as well as Sandra Porter, who's here from the Digital World Biology, and Paul Wilmes from the University of Luxembourg. So I think I'm under 5 minutes, and I'll yield the rest of my time to my colleagues.
So hi everybody. My talk will be about globalization of data-enabled life sciences. And I have 2 slides to show on that. The first one is about the baseline. Where do we stand at the moment, not only in terms of DELSA, but for life sciences in general. So this is data taken in 2010 from the Scopus database on the extent of collaboration in life sciences. The brightness of the lines indicate the extent of collaboration to pair cities around the globe, as well as the extent of collaborations in those 2 pair cities. So if you look at it here, you will recognize the silhouette of certain global regions. So in North America, you can see that there is a good collaboration. Within Europe, Western Europe, there is a lot of collaboration. But as you go towards the east, Asia, there is a real paucity of collaboration. And if you remember in big data, we discussed that creating the commons is the name of the game, and collaboration is also the name of the game to construct these commons. And if you look at South, Africa is almost absent, although things are changing now with the investments in genomics with H2V Africa project in Africa. So this is very important because it tells us the lay of the land at the moment in terms of life sciences in general. It doesn't tell us about data-enabled life sciences per se, but obviously if you don't really have any collaboration, then that's also an area where DELSA Global can really make a concrete contribution. So I think in terms of areas, Asia, Africa, South America, and pretty much many parts of the world are open for collaboration. Now, this is the baseline state. How do we go about being global then? This is a word commonly used, but often misunderstood or poorly studied, in fact. The usual model of globalization actually comes from a fellow who developed the linear model of innovation, Ogburn, in the 1920s. He was tasked by the US government to study innovation, and he came up with this model. It was also the time of Great Depression, so there was a lot of interest about innovation at that time as well, not only at this current time. So his model was sort of the science push model. You have the discovery, and then the people are just supposed to use that, and the society sort of adapts to those innovations. So that's the science push model. And as scientists, many of us really knowingly or unknowingly, subconsciously adhere to this model. We also know that this science push model doesn't work that well. Just because you have a discovery doesn't mean that it will become global or it will become viral for that matter. What is really needed is to engage the end user communities. And with that, I mean, for instance, knowledge users, not only the designers, but also the user communities. So those include, for instance, citizens, small artisan labs, rural communities, who are not necessarily here. But on the other hand, if you remember that DELSA Global really came into existence very rapidly, with strong interest in the last 18 months or so, one can envision that perhaps in the next year or 2, we'll have an even more diverse community here. With diverse, I mean in terms of professional backgrounds, but also under this taxonomy, meaning knowledge producers, designers, and users, where we can also engage with a large variety of user communities. And that will be absolutely important. And also tools, social media tools that will enable those kind of engagement will also be decisive in terms of becoming global. So I'll stop here and move on to the next talk, I think.
Okay. So one of the projects I got involved with this past spring, I guess, was the concept of matchmaking. Okay, so one of the things that's important in community building is forming collaborations, forming networks of people, and doing interesting science. But how do people find their way into these communities who might have interesting ideas and interesting skills and talents to bring to the table? So if you go to Google and type matchmaking, there's really 2 choices. You get Hello Dolly or Fiddler on the Roof. The problem with both is neither is particularly scalable, you know. So we know Gene is very good at this, and we need more of him, but we also need maybe some ways for people to come to a site and find out what is available. And so you could do that. You could put up a website. We have one. We have the DELSA organization. But why would scientists actually use it, right? What would bring people to a site? And that is, you know, if they could come someplace and they could learn something they didn't already know. And so today, you know, we're all very familiar with doing searches on Google. It's the way Larry Smart diagnosed himself, right? So Google and PubMed were his two tools, but he also has an immense network of people willing to help him. He's built a giant team that is pretty distributed because he has an interesting problem and he's an interesting person. So you can see there's, you know, many kinds of resources that one could bring together, but you don't want to have them just as a disconnected collection of objects that you go to this site and do a search. Now I want to go to Vio and maybe find some people. I want to see what's going on in the quantified self. What if instead you could type a term into a search box and it could actually go out to multiple sites and pull together an aggregate of data that you could maybe assemble on a web page, much like you would use your iGoogle form or some of these others, but it would have a scientific focus. And so an idea that we put forth is having DELSA Hub, where you could come in, you could type a query, you could get the state of the art for a given field, and you could find new friends who are doing the kinds of projects and things that you might be interested in working on. And if we built this as a web tool, a resource, it might be a way that scientists would then start to use this as a first place to go instead of simply going to Google, where you get a random collection of things. And in our meeting today, actually, Jeff was talking about, you know, having a different kind of Google, you know, because you can't get into a database with a Google search. You can just get an aggregation of what exists on the web. So, is that me up, or you planning my time? Okay. But anyways, that's what I wanted to share. So I think it's an interesting idea that's worth exploring and talking about in terms of how we get the general community interested in what we're doing and start to learn about the things that are going on in the world. All right. Thank you.
So I wanted to say hello to you all and introduce a new organisation like yours. So this is something of interest to everybody, since we all have chromosomes. But HUPO, their science project, is the Human Proteome Project. And one arm of it, we decided to take on the task for characterising the human proteome and to study the proteins that are missing. We are motivated by a simple hypothesis, which is if we knew more, we'd do better. So it's a huge job. I think there's something like 5 million protein isoforms expressed at one time or another. So it is, as I've been interested in other speakers, challenging to organise global science. And so the HUPO leadership came up with a model looking for national teams or groups of, in several countries, to volunteer for a chromosome. Now, of course, it only makes sense if you study all 24, so we actually ended up with 25 because Italy decided to do the mitochondrial chromosome and the effect on diabetes. So at the Boston meeting, which was in 2012, we really put the whole group together. So we are a very young organisation. And it works quite well in some countries. In some countries, having a national program around a chromosome allows you to attract government funding. Not so much in the US, but I remember the HUPO organisation wrote a letter of support for the Russian chromosome to Vladimir Putin, and that went down well. Out of the 25, it's interesting, and perhaps it represents where we are as a global community. Half the chromosomes are in Asia, 6 are in the Americas, including Brazil. We really are trying to be as international as possible, and then 7 are in Europe, including the Italians. So it gives us then a structure to interact. Of course, when you do a proteomic study, you're generating data on all chromosomes. So now, this is the outline, and I just wanted to show you one more slide. So how do we define a missing protein? It turns about 1/4 of the human proteome is missing. Now, I'm talking about just the major gene product of protein coding genes, not all the minor variants. So about 25% is missing. So, of course, one of the problematical issues is, so what is a missing protein? So the first level of information is no quality MS identification. And one of the challenges is that being a worldwide organisation, labs have different experimental protocols. And so while we do try and get standardisation, technology continues to improve. So the only way it counts is it gets into one of the 2 global repositories which have a very strict curation process, discarding about 1/3 of the data sets. And that's Eric Deutsch's Peptide Atlas in Seattle and Ron Beavis in Manitoba now, the global proteome machine. So they have to be good quality mass spec. So we've begun the job. As I say, about 25% are missing. The next level is no antibody information. Now, of course, I'm not talking about an antibody that's generated to a peptide sequence in a Western blot. This usually is not particularly useful information. So we have an arrangement with Protein Atlas folks, which have very good funding, that the antibody to the missing protein is sent to them and they do their tissue and subcellular localization. Then it's counted. And then, of course, the final point is to try and impute the actual function of the missing protein. So it's a big job, and like you, we have goals. We have a 10-year goal to characterise the missing proteins. So I wanted to introduce the chromosome mission to you all, and I can see some good opportunities to work together. And I think as a species, amazingly, we do so well when we know so little. Thank you.
I'm here today to tell you about the 3D Virtual Cell Project. The NSF funded a grant for us for a conceptual design of an S212 software institute. And the project is led by PI Phil Bourne out of UCSD, and co-PI is sitting right here in front, Gene Kolker. And let me get this. What? I don't want animation. Sorry. I want slideshow. Got it. Okay. Sorry about that. In addition to the stop laughing at me. In addition to the two co-PIs, we have a steering committee of about 14 scientific leaders around the world. We have a community of leading scientists, and we have collaborating organizations such as DELSA, NIF. I don't know where Jeffrey is, but we're doing some collaborating projects with NIF as well. Phil had hoped to be here. He sends his apologies. He has some personal issues that he has to deal with, and he's cut his travel for a while, so he couldn't be here. He did have a recording, but it's 4 minutes long, so I couldn't play it and talk to you. It is on our website, though, if you go to our main page, and it's a 4-minute video explaining the details of the project. The project started out being named the Institute of Translational Systems Biology, and I think it still is, but the biggest project that came out of it, and our branding is now 3DVC. So the focus of the project is to advance the field of cell biology, and I'm going to read this because it doesn't come out very easily for me. I'm not a scientist, I'm a project manager. To advance the field of cell biology through open, community-based science and technology innovation, shared infrastructure, and new sustainable business models. That's a mouthful. What's come out of this since we got the grant was that we've identified that the 3D Virtual Cell Project, and it's become known around the community as 3DVC, and it's to gather a community of scientists together around a shared virtual resource of creating the actual three-dimensional virtual cell. So the first thing that we did, oh, I don't want him to start talking. Oh no, I promised, Gene. Somebody help me turn this off. Don't start talking. There, okay. That was Phil. And you can see him if you go on the website. The first thing that we did was we actually held an amazing meeting in San Diego, and 170 scientists got together and from around the world, and they explored the grand challenge questions surrounding whole cell modeling. Specifically, what obstacles might there be to meeting this goal of creating a 3D virtual cell? After 44 TED Talks, 2 days of workshops and brainstorming similar to what we did here today, there was a general, and I say general, I think we probably wanted it to be more specific, but a general direction for the project came out of that, and we ended up with massive amounts of things that the community wanted. And I think the community as a whole, or at least the 170 that were there, thought that this is a project that they wanted to get their hands around. It's since grown to over 400 people who have contacted us to participate in this project. So the next thing was we had several small workshops, work groups that got together to look at what came out of the conference. And it was clear that the community wanted a hub, and we've talked about that over and over again here today. Some sort of place where they can coalesce, they can work together on projects. So we're in the middle of changing our website, which was informational-based, to a collaborative website. We're building it ourselves on Drupal, but we're using NSF's EarthCube, Ning, as an example. Another one is Commons in a Box, CUNY Commons, and they've got some pretty nice project-based systems, which we're putting into place right now, where our project teams will be able to work together, upload files, collaborate together. So that's in the works. In fact, if you look at the website now, it's partially up. The other thing that we hope to get out of this by putting this community together is to get some input and clear understanding of what's needed to answer those challenging questions. I mean, like, what is a 3D virtual cell? You know, it's like, is it going to be a generic cell versus a specific cell type? Is it an actual cell or a modeling toolkit? You know, these questions haven't been answered yet by the community. So it's going to be a cellular information framework. Another thing that came out of the conference was that there was the need to bridge the gap. So how do we catalyze more effectively effective change between theoretical computational scientists and the experimentalists? And that came up over and over, so we're going to be addressing that as well. The other thing that came out was that we needed to inventory what's out there in the cell biology, what tools, so we're doing a collaborative project with NIF, which is the Neuroscience Informatics Network. And we've been building a catalog on top of their resources, and we're building a front end on our site, and it will have data sets, models, software, education tools, media, viz tools, and we're just about to launch that probably in the next 2 weeks. That'll be up. And it'll have an opportunity for our community to be able to add tools to it, which will populate the NIFT data resource. Is it really over? Oh my God. Okay, so one more thing is we're going to add a model repository. We're in the middle of designing that, and we'll be working with DELSA on that. And then we're collaborating with force11.org, which is, God, how did that go? Part of that was the mess up here. Yeah. So I get another minute.
Yeah, 20 seconds.
Okay. Force11.org is the future of research communication and e-scholarship. I encourage you all to look at that. We're doing some pilot projects right now on standards and sharing and evaluation. All of these tools will help us lay the groundwork for a software center proposal. And thank you very much.
The reason I am presenting this one is we had a recent exciting development at DELSA Global on microgrants. We won't get into details now, but I would like to introduce the concept of microgrants and citizen philanthropy, because I and we think that it's directly relevant for the future development of DELSA Global. But before we go any further, this is also about the science of hows. How do we do science? Because science has usually been about science of what's. In other words, which molecule did you discover? Which biomarker did you discover? But equally important is how do you do science? And one of the upstream elements in science is really research funding. There comes a point, or there might come a point, that you may want to take the matter to your own hands. In other words, one option is to write grants and apply for grants and ask money from other people, which is fair. We all do that, and I think we'll keep doing that. There is another option, though, that we may want to raise the funds ourselves. So there has been a number of trends in terms of how we do science in 21st century science, and this is not just old wine in new bottle. There are really new ways of doing science. So one of them has been crowdsourcing, crowd analysis. The latest crowding is in crowdfunding. And if you think this is sort of small change, that's actually not true. Crowdfunding is booming, and it was 2.7 billion last year, with an increase of 81% over the prior year. And it's estimated that it will be 5.1 billion this coming year. The crowdfunding report was just recently released on April 8th. So we're talking potentially large sums of money, actually not potentially, large sums of money that can be utilized in a versatile way. So what I want to discuss today is about opening up the design, idea generation, brainstorming phase of science. Because if you think of the innovation trajectory, you come up with an idea, not necessarily subscribing to a linear model, but essentially we need an idea. Then you implement it, test the hypotheses, and then you interpret. So this is a common framework. So in the top left, you see the classic model. You have the principal investigators. They come up together, usually a couple investigators. You bring your confidants, trusted friends. Usually that's how it works in practice, and you put out a proposal. So that's this part. But everybody is working individually in isolation. And then there's a panel of experts that are trained in a similar way. They might be also competing with you in different ways, and you have to pass them. And those very few ones who are lucky enough who can pass through science, technology, and politics, then they are actually awarded, and they do, with small teams, working in small teams, they do some projects. So this is the classic model, and this has been somewhat prevalent over the last 400 years or so, since the start of the Enlightenment. But what happened is that we are sort of accustomed to this idea that we now take this as a given, as though we have no other options. So I will argue today with you that, or I'll submit to you that there might be other options. And there are several reasons for that. We're in the age of big data. In the age of big data, experts, or the classic PhD or MD experts, simply do not have the muscle power to mine or valorize the big data to innovation. We need more than just the experts to work on big data, whether it's generation of ideas, analysis, or valorization, or ascertainment of utility of big data. So that's number one. Number two important point there is that experts often have blind points. In other words, they are not always engaged with user communities, and we find that the questions that are asked by experts may end up being not relevant when the product actually hits the market or the clinical practice or public health for that matter. So in the alternative model, what we propose to do in the lower right-hand side, the design or idea generation phase is also opening up both among experts, but also citizens, user communities, patients, or future patients, if you like. We had a keynote presenter, Larry Smart. So individuals who are interested in health in general, but those who may not be directly interested, but simply citizens, then come together and generate the ideas. And it is in this stage that we propose this concept of microgrants. So these are not Kickstarter funds, but rather prior to that, they can be even in the amount of $1,000 or $2,000, that will allow a number of citizens to come together with experts, to generate ideas. So this is a very novel idea in itself in terms of how we do science, because generation of idea has usually been about sort of a cloistered activity, and so this can open up the sort of this design space. And also peer review is also opening up in that we don't only bring the classic experts, but also user communities to the peer review process, and then we generate a number of ideas, and they're assigned to larger teams. And there can be a range of microgrants and crowdsourcing where you can generate larger sums of money to actually enable those larger teams. And interestingly enough, that's what DELSA Global has been doing over the last year and a half with a number of projects. And today as well, we generate a number of ideas in an open manner, nobody withheld their ideas, and then we generate the ideas. And then we also have a large, diverse group of panel here, people coming from all directions, and then we generate a number of DELSA-endorsed projects. So I will stop here and just want to alert all of us that there are other ways of doing science, perhaps more gentle and more collaborative ways of doing science, where everybody feels empowered, where everybody feels creative. But most importantly, in the age of big data, coming back to the original point, we do two things. We have the muscle power of the masses to mine the big data, to link that to innovation. And secondly, we can also remedy the professional blind spots that we all tend to have. Thank you.
Thanks to all our speakers. We lost Bill. I think he had to leave early. I can open the floor for questions to any of the speakers or comments.
So whether do I know them? Well, there is a lot of examples, and it's hard to know all the crowdfunding in a way that's consistent with the model. So any community can come up with this, and but it's really booming, and that's really an alternative model. If you have an interesting idea, or you can actually generate the idea. Oh, I'm sorry, that's right. Now it rings the bell. Of course, I have colleagues in Israel, in Tel Aviv, who have, it might be the same institute actually. I met with them a few weeks ago. So they are very interested in this, and they are working in crowdfunding. And it used to be, and I think rightly so, with rare disease communities, because they're isolated and they need each other. But then you can also argue that we're also isolated in big data community. Everybody's generating big data. There are silos. And I will argue that, you know, we're all isolated, and to the extent that that's true, we need each other also for crowdfunding. In other words, shared problems require shared solutions.
That's a model that I'm personally familiar with, and it works very similar, and it's working very well, actually. Jimmy Lin, he pioneered that model a few years ago now. The model is bringing together scientific experts in specific disease areas, as well as in genomics and in informatics. So he puts together a virtual team, geographically distributed, and a clinician who recruits patients into the program, for whom they generate funding through crowdsourcing, and then fund that research. So it's working well.
Any other questions? Any comments from the panel?
I think we're kind of getting tired a little bit. So I have one general question for all of you guys, if that's okay. So you touch at very important, issues, how to do science differently, how to accelerate what we do. What do you think would be doable within 1 year? So you're going to see it in a year from now, maybe in the same place, maybe close to it, right? What do you think is doable with regard to sustainability of science, with regard to what, Jack, you were talking about? What do you think would be doable with regard to quantified cell? And then, basically on your topic, each of your topic.
With or without funding?
I'm assuming some funding. I think within a year, and I'm inclined to talk about science of sustainability. Sustainability of science is perhaps related but different. So the science of sustainability that our proposal addresses, I think within a year we could have a robust network that's highly active and interactive. And I think we could begin to do some of the first things in terms of, in addition to stimulating additional creative scientific activity within the network, we could and should begin to design first round of communications to high school, college, and graduate students to illuminate the importance and the possibilities of scientific careers attacking really important challenges in sustainability. And I think we could have a first round policy briefing for those who need to understand better than they do today the power and importance of this kind of science, and of course the criticality of the sustainability issues.
I agree with him. I think for the 3D virtual cell, in a year, I'd like to see the community have come together with what direction we're going to go in. And we already have a project layout of each area of the virtual cell and team leaders for each one. So I'd like to see those teams active and working together. And of course, collaborating with DELSA.
Just to have a direct answer to Eugene's question, in 1 year, I would assume that in a burning area for data-intensive and big data science, DELSA Global presumably could champion a crowdfunding on a global scale, given also coupling it with our international outreach. So that's one tangible answer. The second thing, I think this kind of approach, collective innovation, is not to make everybody the same. If you look at human history, that's a more sociological, philosophical look at things, we have two sort of interests, a sense of exploration, but also a sense of belonging and a sense of community. And we need both. So in other words, we need our autonomy, individual agency to be creative, but also we want to belong to something. So in a very competitive environment, we tend to lose the sense of community, but yet we don't want to be entirely lost in the community, and we don't want to lose our individuality. So measures or tools such as crowdfunding can strike the right balance, so we are autonomous, creative, but we also have a sense of belonging. And I think that's the basis for 21st century sustainable science, to tie that to Jack's, because it's not only about technology, but the values that drive that science. And it's a hybrid value now, both competition and cooperation.
Oh, I got my own. So I'm going to remember what you said earlier, that we underestimate the progress over a long term and overestimate it on a short term. So in a year, so I think in a concept like a DELSA hub, and using it as an interface to pull together a lot of the aspirational things we've talked about, I think a few key resources, and you could measure maybe some growth in the DELSA community, maybe membership that from individuals that we haven't contacted, and interest in that. I certainly don't want to get into quantifying numbers, because that would be hard to do. But you know, I think we could find some measurable growth, and I think that would be very important, and it would be very important to the to some of the missions that have been already laid out.
On a similar note, I have a question regarding what you think DELSA's role in each of these projects. Some of them are really immersed in DELSA and it started here. What is DELSA's role in each of these projects that couldn't have been achieved without the community behind it? It's kind of hard
I like
questions
starting at
here.
that end. That's a Jack question. Yeah, we all voted.
I literally could not envision the proposal that we are making without the predecessor existence of DELSA. So it's really hard for me to answer that, other than to say that it is organically a precondition and a kind of a launch mechanism for the research network that we would like to put together.
I think,
That's a Phil question.
well, I mean, I think we're in the development stage of the community itself, but we're going to be adding a model repository and some ability for our community to upload and download models, and I can see DELSA helping with that. And then I'm already in talks with Courtney about how we're going to integrate our community with your new matchmaking community. So we're already in talks to make sure that we stay connected. And part of the 3DVC is we're always looking for other groups that are already building something, already doing something that we could, you know, piggyback on instead of reinventing the wheel. And we've done that with NIF. We're doing it with DELSA. There's another group that we're looking at called Index that's not up yet, but will be coming online soon. So it's a huge benefit for us.
I think DELSA's role is really as an alliance. DELSA is not a society, it's an alliance. So in other words, it helps govern the big data and technology challenges. And governance is different than government. When you have global technology, we can't really control or regulate it, but we can govern it, where we will always have uncertainty. So by having a rather relatively loosely associated group of transdisciplinary individuals, we can govern technology. So I see, in my view, DELSA Global is a governance tool to steer innovations as they emerge and diffuse.
Yeah, and along those lines, I think, you know, some of the themes we've heard earlier today, and you know, there's the technology of technology, and there's a lot of smart people in this room, a lot of really good ideas, but then there's the social people issues around technology adoption, use. I think, you know, we've spent a lot of time actually at this meeting on human health kinds of data, and these are going to be very, very interesting challenges. And I think, you know, DELSA can play a role in that conversation and guidance and formulate some ideas.
Okay, I have an afterthought, just one. If DELSA didn't exist, it would be necessary to invent it.
Yes, you did. um, so I actually think this is a really important point, that this is an alliance and it's something to bring back home for us, because I get asked what DELSA is and I have to think about for a minute before I answer, but I answer in a similar way, that it's a community that's growing. So I think that was really obvious from the comments, that this is a voluntary alliance effort, and the difference from others is I think that we have all kinds of people here, all kinds of expertise, and it makes it different from other standardization efforts for me. We have a question here.
Hi, this is a question for the 3D virtual cell folks. When I was studying fatty acids many years ago, there was actually no basic biology review paper on, there are 300 different types of cells, and there was nothing that said, here's how all these cells develop, mature, and die. My question is, where are you going to start?
You're in what?
Where is the effort going to start?
Gene?
Gene?
It's already started, and
Yeah.
we started not by answering one specific question, but basically like we did start DELSA. So Phil and Stephanie invited people, invited community to come together and think about how it can be done, okay, in principle. That's conceptualization award we got from NSF. So we kind of put together those transdisciplinary expertise people, teams, okay? And we wrestle with those ideas, brainstorm what can be done, how long it will take, what technologies, what approaches would we need. So that's kind of background, okay? And right now, I think this catalog of resources, right? This is a major important point, because we did overview, lot of, I don't know, we started what, 2,000+, right?
Yeah.
It was ridiculous number of potentially relevant resources. And now we're down to 200 or
Actually, it's more than that now.
more than that. Oops, I thought we did 10x
Well, and-
reduction.
Yeah, and
But-
the other thing too is, to answer your question, is that came up a lot at the conference. Which, do we start with a yeast cell? Do we start with some easy something and start there? And then someday we'll end up with all the cells maybe, but there was a lot of conversation and the community needs to decide, because, if you start with a yeast cell, then you eliminate a whole bunch of other scientists. So they're talking about that. Then if you want to put input, join the community and go to the website and join.
I have a question that may be obvious to all of you. I'm sorry I had to miss, I was in and out because of meetings, sequestration and all. I was wondering if you actually have a profile. I mean, that picture kind of shows a profile of a population. You have a profile of DELSA. Who is in DELSA? Because your biggest resource, I think, are the people who are part of it. Is there a profile that you have, or can you create a profile? It comes to all kinds of demographics. And I think that my goal would be, if you find out that most of you are extremely brainiacs, IT, mathematician, physicists, and so forth, maybe you need to increase more the population of people who would love to use your data, would love to understand what is possible. I've been in that role for all my life, where I try to translate between people who see equations and get excited and people who go like, I can't see anything like it because I'll run away. So that's what I'm trying to make sure, that you have the right mix, because people who know biology have great questions, but they don't even know what they could ask because they don't know what are the tools. People who have the tools know a lot about the tools, but are not really sure what questions would be most relevant. So the hammer and the nail.
That's a great question, though you definitely missed most of our presentations today. Just look at this, people who
No, but could you make like a term cloud from your abstract or something?
We have to do that, you're absolutely right. But if you can just...
Can I quickly
Can
answer?
you guys tell your expertise? What your origin, kind of coming from?
I'm a sociologist by training.
I'm a project manager by training.
I'm a scientist who moved down to social studies of innovation.
I like that. So, yeah, scientist by training, and have wandered many different disciplines through my career, so.
I'm a geek.
In the room, yeah.
What was the question?
What was the question?
How many biologists do we have?
How many biologists do we have?
Yeah, we've got a few. No? How many IT?
IT?
Computation, how many bioinformaticians?
Eugene or Ilkay, can I quickly answer the prior question? That's an important question, actually. Who is the DELSA community? That's really a taxonomy question. Who is the community? How do you define this taxonomy of DELSA Global? There are different ways of going about it. One is the doers of science. So some call this actors of innovation. So they are scientists, people who engage in knowledge generation, and we have a variety of them across different disciplines. So those are actors. So that's taxonomy dimension one. The second one are the narrators. So we have actors and narrators. In a way, DELSA Global is a narrator because it's examining the innovation and see how actors are coming together or not. You need the narrators to steer the innovation, and that's how you build the alliance. So we need a very transdisciplinary, sort of pan-disciplinary distribution, as well as narrators. So some social scientists do that. And that's the taxonomy of DELSA Global.
This is the last question, and there's one more announcement before I lose everyone. If you have more questions on Larry Smart's presentation, Jerry will be in the sort of close proximity, I think, during the drinks.
I did call in this morning, so I can answer any question. Possibly would have more of any of the data.
There you go. Last question.
So this is just to follow up on the discussion. We spend a lot of time text mining PubMed, and we build up concept profiles for genes or diseases or proteins. But part of the PubMed abstracts that we mine are people. So we've also built concept profiles for individual people based on all of the things that they published about. So we have a list of, I think, 1.7 million people from PubMed with their concept profiles, and we've been planning to do exactly what I see in this lower panel, which is if you do some kind of text mining experiment and you have as an outcome some network of genes, proteins, processes, diseases that no one expert's going to know about, that we would try and locate the expertise we would need to interpret the graph that came out of the data set. So that's maybe one way to look at it.
Thank you. Let's thank our speakers and call our leader back.